Welcome to Bio Bits and Bytes. Today we're taking a really deep dive into the world of bacteria and how they share genetic information. We're talking genetic transformation and plasmid transfer. We got a lot of research in front of us, and well, the mission today is to unpack how bacteria grab new genes, from their surroundings or even through direct contact. It's a whole hidden world, but with some pretty big consequences for us.
Absolutely. These processes are fundamental to how bacteria evolve, how they adapt. And yeah, big consequences, especially when you think about things like antibiotic resistance. And for everyone listening, don't worry if some terms sound complex. We'll definitely break down the key scientific ideas and explain how these mechanisms actually work as we go.
Great. Okay, so let's start right at the beginning then. Genetic transformation. What exactly is that?
Right. So genetic transformation, in simple terms, is when a bacterial cell takes up genetic material, usually DNA, directly from its environment. This external DNA then gets incorporated, becoming part of the bacterium's own genome.
So it just absorbs it.
Yeah, kind of like absorbing something, instructions, you could say, from its surroundings and making them its own. It directly changes the cell's genetic blueprint.
Okay. Now, I know the word transformation gets used elsewhere, like in cancer research. This is the same thing. Just want to be clear.
Ah, good point. No, definitely not. It's a completely different meaning. In cancer biology, transformation means a normal cell turning cancerous, becoming malignant. Here in bacteriology, genetic transformation is specifically about this uptake of foreign DNA, nothing to do with malignancy.
Got it. No microscopic bacterial tumors then, just grabbing new code. So how long have we known about this? It sounds pretty fundamental.
It goes back quite a way, actually, to 1928 and a scientist named Frederick Griffith. He was working with Streptococcus pneumonia, the bacteria that causes pneumonia, trying to make a vaccine.
With heat-killed bacteria, right? That was the standard approach.
Exactly. But he stumbled onto something really strange. He had two strains, a virulent one which caused disease, and a non-virulent one which was harmless. He found that if he mixed heat-killed virulent bacteria with live harmless bacteria and injected them into mice, the mice got sick. The harmless bacteria had somehow become virulent.
Wait, the dead ones made the live, harmless ones dangerous?
How? That was the million-dollar question. Griffith didn't know what was being transferred, but he knew something was. He called it the transforming principle.
The transforming principle. So he named the phenomenon, but not the molecule responsible.
Precisely. That breakthrough came much later, in 1944. Oswald Avery, Colin MacLeod, and Macklin McCarty, they did this really elegant series of experiments. They took the virulent S pneumonia, carefully isolated different molecules, proteins, RNA, DNA, and tested each one. And they found that only the purified DNA from the virulent strain could transform the harmless strain into a killer.
Just the DNA itself, that must have been huge. Proof that DNA carried the genetic information.
It was huge, revolutionary even. But, the scientific community was skeptical at first.
Really? Why?
Well, the prevailing thought at the time was that proteins, being more complex, were more likely candidates for heredity. DNA seemed too simple to many. It actually took the discovery of other ways bacteria exchange genes, like conjugation in 47 and transduction in 53, both thanks to Joshua Lederberg before Avery, MacLeod, and McCarty's work showing the transforming principle was really widely accepted.
Wow. So even groundbreaking evidence needed backup from other discoveries. Okay, so bacteria can take up DNA. Do they do this all the time? Or are they constantly sampling their environment?
That's a really good question. No, it's not usually a constant state. Many bacteria need to enter a specific physiological state called competence.
Competence.
Yeah, it's a temporary state where the cell is actually able to bind and take up DNA from the outside. Think of it as opening the gates for a short period. And interestingly, we can actually trigger this state, induce competence in the lab using chemicals or even electricity.
So you can force them to become receptive in the lab. But what about out there, in nature? What makes them decide, okay, time to become competent?
Natural transformation is thought to be ancient, maybe going back to the earliest prokaryotes. And a key driver seems to be DNA repair.
Repair? How does taking up foreign DNA help repair your own DNA?
Well, imagine a bacterium's DNA gets damaged, say by UV light or some chemical. If it takes up DNA from a neighbor, maybe even a dead one, that DNA might contain an undamaged copy of the gene that got broken in its own chromosome. It can then use that external DNA fragment as a template through a process called homologous recombination to fix its own damaged sequence. It's like finding a patch.
I see. So it's a survival mechanism, a way to maintain their genome integrity.
Exactly, survival and repair. But it's also a source of new traits. Taking up DNA can introduce entirely new genes, leading to genetic diversity and adaptation. Acquiring antibiotic resistance this way is a prime example.
Right, so repair and innovation, essentially.
Right.
Which bacteria are known to do this naturally? Is it common?
Oh, it's surprisingly widespread, especially among medically important bacteria. We see it in gram-negatives like Helicobacter pylori, the stomach ulcer bug, Legionella pneumophila, Neisseria meningitis, Neisseria gonorrhoeae, Haemophilus influenza, Vibrio cholerae.
Wow, a lot of familiar pathogens.
Indeed, but also in soil bacteria like Pseudomonas stutzeri, Acinetobacter bailey, plant pathogens. And among gram-positives, you have Streptococcus pneumoniae again, Streptococcus mutans involved in cavities, Staphylococcus aureus, Bacillus subtilis from the soil. It's been reported in at least 30 species just within the Pseudomonodonta phylum alone. So yeah, it's a common strategy.
That was a huge range. Okay, so what kind of conditions actually trigger this competent state naturally?
Often it's linked to stress for specific growth phases. Things like high cell density crowding or running out of nutrients. These often happen when bacteria enter the stationary phase, when growth slows down.
So when times get tough, they might look for external solutions.
That seems to be the logic, yes. It varies a bit, though. Haemophilus influenza is most competent late in its rapid growth phase. Many streptococci, like Streptococcus mutans, ramp up competence at high density, often linked to forming biofilm.
Biofilms, right? Those sticky communities.
Exactly. Bacillus subtilis often becomes competent when starved for amino acids. Micrococcus luteus, too, during late growth or amino acid starvation. It really looks like they're sensing their environment and adjusting their ability to take up DNA accordingly.
It's like built-in sensors triggering this genetic uptake ability.
Hmm.
You also mentioned something really interesting earlier, that DNA damage itself can induce competence.
Yes, that's a key finding. In many cases, the very agents that damage DNA also flip the switch for competence genes. For instance, in Streptococcus pneumonia, mitomycin C, which cross-links DNA, and fluoroquinolone antibiotics, which cause double-strand breaks, both make the bacteria more likely to take up DNA.
So the damage itself is the signal.
It seems so. In Bacillus subtilis, UV light boosts transformation. In Helicobacter pylori, ciprofloxacin, another DNA-damaging antibiotic, enhances transformation. It's like the cell senses the damage and thinks, okay, better open the doors and see if there's a repair template floating around.
That makes so much sense from a survival perspective.
Absolutely. Work on Legionella pneumophila really hammered this home. They found that only toxins that actually damaged DNA strongly induced competence. This supports the hypothesis that competence really evolved at its core as a response to DNA damage. Even Deinococcus radiodurans, that incredibly radiation-resistant bacterium, uses natural transformation for DNA repair under stress.
It paints a picture of transformation being fundamentally about maintaining the genome. Okay, let's switch gears slightly. How do scientists harness this in the lab? You mentioned inducing competence.
Right. Lab transformation is a cornerstone of molecular biology. For bacteria, there are two main workhorse methods. First, chemical transformation. This usually involves treating the cells with chemicals, classically calcium chloride, sometimes combined with a heat shock. This makes the cell membranes leaky, more permeable to DNA that you add to the solution.
So chemicals to make the walls temporarily leaky. What's the other method?
Electroporation. This uses a short, high-voltage electrical pulse.
Zapping them.
Basically, yes. The pulse creates temporary holes, or pores, in the cell membrane, allowing DNA from the surrounding buffer to zip inside before the membrane reseals. It can be very efficient, though sometimes a bit harsh on the cells.
Okay, chemical leaks or electrical holes. Once the DNA is potentially inside, how do you know which bacteria actually got it? There must be millions that didn't.
That's where selection comes in. Usually, the DNA you're trying to introduce is carried on a plasmid, a small circle of DNA, and you design this plasmid to carry not just your gene of interest, but also a selectable marker gene. Very often, this is an antibiotic resistance gene.
Ah, so you add an antibiotic.
Exactly. After you do the transformation procedure, chemical or electroporation, you spread the bacteria onto an agar plate containing that specific antibiotic. Only the bacteria that successfully took up the plasmid and are now expressing the resistance gene can survive and grow into colonies. Everything else dies. It's a very effective way to isolate the transformed cells.
A neat trick. Survival of the transformed. But what if you need to know if your specific gene of interest is there and working, not just the resistance marker?
Good question. That requires screening. Sometimes the resistance marker is the gene of interest, but often it isn't. So you might include a reporter gene on the plasmid, something easy to detect. Green fluorescent protein, GFP, is a classic. Transformed colonies glow green under UV light.
Like tiny little beacons.
Precisely. Or luciferase, which makes cells produce light if you add the right substrate. Or you can use molecular methods like nucleic acid hybridization with probes to directly detect your DNA sequence, or immunological methods using antibodies to find the protein your gene makes. Lots of tools for screening after the selection step.
Okay, selection finds the ones with the plasmid, screening finds the ones where it's doing what you want. Makes sense. Now, we've talked a lot about transformation, taking up DNA from the environment. But you also mentioned plasmid transfer via conjugation, direct contact.
Yes, conjugation. This is another major route for horizontal gene transfer, but very different from transformation. It requires direct cell-to-cell contact. It's often called bacterial sex, although it's not really reproduction, more like a direct donation of genetic material.
Bacterial sex.
Yeah.
Okay, how does that work? Do they shake hands?
Sort of. The donor cell, which has a special type of plasmid called a conjugative plasmid, grows a structure called a pilus. A sex pilus, actually.
A pilus, like a hair.
Exactly. A thin protein appendage. This pilus extends out, contacts a suitable recipient cell, one that lacks this plasmid, and then it usually retracts, pulling the two cells together.
Okay, reeling it in.
Then a channel or pore forms between the two cells, a mating bridge. The conjugative plasmid in the donor gets nicked. One strand is cut, and that single strand is actively transferred through the bridge into the recipient cell.
Just one strand.
Just one strand initially. While it's being transferred, the donor synthesizes a replacement strand using the remaining circle as a template. And once the single strand arrives in the recipient, it also gets replicated to become double-stranded. So at the end, both donor and recipient have a complete copy of the plasmid.
Wow. So the donor keeps a copy and the recipient gets one. That seems very efficient for spreading the plasmid.
Extremely efficient and incredibly important. Conjugation is a primary way bacteria share advantageous genes, like those for antibiotic resistance, virulence factors, or new metabolic capabilities. It happens everywhere: soil, water, inside us. It's a major engine of bacterial evolution and adaptation, and, unfortunately, a key reason why antibiotic resistance spreads so rapidly.
That direct transfer really explains the speed, doesn't it? You mentioned the pilus and the plasmid. The instructions for all this machinery must be on the plasmid itself.
Yes, largely. The genes needed for conjugation, called the tray genes for transfer, are usually located together in a specific region, an operon, on the conjugative plasmid. These tray genes code for the pilus proteins, the proteins that form the mating channel, the enzymes that nick and process the DNA for transfer, everything needed for the process.
Whole toolkit encoded right there.
Exactly. And the expression of these tray genes is very tightly regulated. Making a pilus and the transfer machinery costs energy, and it can make the bacterium vulnerable, so they don't usually do it unless conditions are right, or if they've acquired mutations that cause deregulation.
Regulation makes sense. Don't advertise unless you need to transfer.
Okay.
We have transformation, environmental DNA uptake, and conjugation, direct plasmid transfer. Our original plan also included transposons. What role do they play?
Transposons. The jumping genes. These are segments of DNA that can move from one location in the genome to another. They can jump within a chromosome, from a chromosome to a plasmid, from one plasmid to another, or even just to a different spot on the same plasmid. They're mobile genetic elements.
Jumping genes. That sounds potentially disruptive.
It can be, but it's also a powerful source of genetic variation. By moving around, transposons can rearrange genomes, disrupt genes, activate others, and importantly, they often carry other genes with them, like antibiotic resistance genes.
So they contribute to the shuffling of the genetic deck. Are there different kinds?
Broadly, yes. There are class 1 retrotransposons, which move via an RNA intermediate copy and paste, essentially. But in bacteria, we mostly deal with class 2 DNA transposons. These move directly as DNA. The simplest type are insertion sequences, or IS elements.
IS element.
Yeah. These are typically quite small, maybe 1,000 base pairs or so. They basically just contain the gene for an enzyme called transposase, which is the enzyme that does the moving, flanked by short inverted repeat sequences at each end. The transposase recognizes these repeats.
So just the moving machinery and the signals it recognizes. Minimalist design.
Very minimalist. The transposase enzyme binds to the inverted repeats, cuts the IS element out of its current location, and then inserts it somewhere else in the DNA.
How does it insert? Does it just find a random spot?
It's often somewhat random, though some transposases have preferences for certain target sequences. The insertion process itself is interesting. The transposase makes staggered cuts in the target DNA, inserts the IS element, and then cellular repair enzymes fill in the gaps. This filling in process creates short, direct repeats of the target site DNA flanking the newly inserted IS element. Finding these direct repeats is often a clue that a transposon has landed there.
Okay, so the transposase cuts, pastes, and leaves a little signature behind those direct repeats. What happens if an IS element jumps into the middle of a gene?
That usually inactivates the gene. It's a common way mutations occur. But they can also land in regulatory regions and change how genes are expressed, sometimes increasing expression, sometimes decreasing it. Or they can just add their DNA without much effect. It depends where they land.
So these little IS elements can cause mutations just by hopping around? Are there transposons that carry more than just the transposase gene?
Yes, absolutely. Those are called composite transposons. Imagine a stretch of DNA maybe carrying an antibiotic resistance gene or two that happens to be flanked on either side by an IS element.
So IS elements acting like bookends.
Exactly. The IS elements can be in the same orientation or inverted. The transposase enzymes from one or both of the IS elements can recognize the outermost inverted repeats and move the two IS elements and everything in between as one unit. This is a major way that things like antibiotic resistance genes get mobilized and spread. TN10, which carries tetracycline resistance, is a classic example.
So the IS elements provide the mobility for whatever genes happen to be caught between them. Clever. Any other types?
There are also non-composite transposons. These also carry extra genes like resistance genes, but they don't have flanking IS elements. Instead, they have their own short inverted repeats at their ends, and they encode their own transposes, plus often another enzyme called a resolvase. TN3, carrying beta-lactamase resistance, is a well-known example. They move by a different, slightly more complex mechanism, often involving replicating the transposon during the process.
Replicating it. How does that work?
It's often called a replicative transposition mechanism, sometimes involving a co-integrate intermediate. Essentially, the donor DNA molecule containing the transposon fuses temporarily with the target DNA molecule. During this fusion, the transposon gets duplicated, so you end up with this larger fused molecule that co-integrate with two copies of the transposon at the junctions.
Okay, a temporary fusion with two copies, then what?
Then the resolvase enzyme, which is often encoded by the transposon itself, like in TN3, performs a site-specific recombination event between the two transposon copies. This resolves the co-integrase back into the two original DNA molecules, donor and target, but now each one had a copy of the transposon. It's a copy and paste mechanism, unlike the simple cut and paste of many IS elements.
Copy and paste versus cut and paste. So transposons add another layer of genetic mobility and gene shuffling within and between bacteria.
Precisely. Between transformation, conjugation, and transposition, bacteria have this incredible toolkit for acquiring new DNA, repairing damage, and adapting rapidly to changing environments. It explains a lot about their resilience and evolutionary success.
It really does. We've covered a lot of ground, from Griffith's transforming principle to the complexities of conjugation machinery and jumping genes. It's clear these processes are absolutely central to the bacterial world.
They really are. Understanding the nitty-gritty, how DNA gets in, how transfer is regulated, how elements like plasmids and transposons move, it's fundamental. It highlights just how dynamic bacterial genomes are.
And thinking about the implications, antibiotic resistance is the obvious big one. But it also makes you wonder how we might harness these processes, perhaps for biotechnology.
Exactly. There's potential there too. But certainly understanding these natural mechanisms is the first step, whether we're trying to combat resistance or leverage bacterial genetics for other purposes.
It definitely leaves you thinking about the constant genetic conversation happening in the microbial world. Thank you so much for walking us through all of that today. It was incredibly insightful.
It was my pleasure. It's a fascinating area.
And thank you, our listeners, for joining us for this deep dive on Bio Bits and Bytes.